hopefully my mic works. Yes, hello and welcome back to another day, another lesson. And right now, we are going to talk about a bit more of metabolism, but maybe going into like flavoproteins and stuff like that. Okay, so let me just double check something really quickly. Yep, the mic works. Alright, so we know that amino acid metabolism is complex, right? It's more complex than carbohydrate and fat metabolism because the thing is, it involves not only energy extraction, it also involves nitrogen handling, and to use amino acids as energy sources, their nitrogen must first be removed and the remaining carbon skeleton can then enter, we talked about this earlier, it can then enter the glycolysis or TCA cycle. So some amino acids like lysine and leucine, they are purely ketogenic. So what does that mean? So lysine and leucine are actually, let me just erase this smiley face. They are ketogenic and are converted from like being an L amino acid into an alpha keto acid. So that's what happens to them, and you also get ammonia as a result. Okay. And amino acid oxidases are flavoproteins that catalyze the oxidative deamination, which would produce the alpha keto acids, and they have the important FAD as a cofactor, which would accept electrons from the amino acid substrate, and that would then form an amino acid intermediate. So how that would look is it would end up looking like this. Eraser. Okay, so you have, where is it? If you're talking about FAD and FAD plus FADH2, I mean, you know that it's a cycle, right? But then you have RCH2 NH2. That one goes from itself into an RCH NH2 plus. And then over here it goes from oxygen to water. So that's what happens to it. The oxygen is reoxidized. The oxygen will reoxidize FADH2, which would then release H2O2. And take note, ammonia is very, very toxic and must be safely transported and eliminated. So the way that we get rid of that is it is incorporated into glutamine by glutamine synthetase in an ATP dependent reaction, and glutamine serves as a non-toxic carrier of ammonia in the bloodstream. And once in the liver, glutamine actually is hydrolyzed by glutaminase, which would release the free ammonia, and that can then be used to generate the carbamoyl phosphate for entry in the urea cycle, as mentioned earlier. Okay. And CPS1, what is that? You have carbamoyl phosphate synthetase, okay? It catalyzes the first step, so the first and rate limiting step of the urea cycle. So if you want to see how that happens, it goes like this. Let me just erase this one. Okay, so we have ATP plus NH2, whoa, plus carbon dioxide into, whoa, wait, no, that we have to make sure that we write this end part properly. Go and become an double bonded oxygen and H2O over here and a P. Nice, beautiful, thank you. Welcome. And this diagram illustrates the regulation. Oh no, I have yet to draw that one. So you have the regulation and operation of the urea cycle. So how that happens is, okay, yeah, we have to try and write it out. The pencil might be too thick, but if I make it, oh no, that's the eraser. So you have here CO2 plus NH4 plus, right? That one, you get the CPS1, the carbamoyl phosphate synthetase 1, and carbamoyl phosphate is the result. And then it would be converted, like ornithine transcarbamylase would help and make it into citrulline. And where is citrulline? That is like in the cytoplasm, like from citrulline to the arginosuccinate, and then arginine, and then ornithine, and blah blah blah. So that is the urea cycle. In the real world, our version of magic is science. Yes, exactly. Science. Wait, is science with sparkly. Yay, I agree. IRL magic is science. And yeah, you have one carbon metabolism as well. It involves the transfer of single carbon units in various oxidation states. So we know that we have methyl and methylene, right? So if we erase this, handwriting is so beautiful, it's like art. So you have methyl, CH3, methylene, that is CH, because it's an ene. And then you have formyl, H3 double bond O, and the most reduced and simplest form is the methyl. And the methionine, an essential amino acid, would serve as a precursor for the S-adenosyl methionine, the primary methyl group donor, which is used in the different methylation reactions across the cell. Yes. And you have a couple of amino acids which has sulfur, and those are like for different roles. So the sulfur amino acids are, let's see, wait, where's my marker? Okay, so you have cysteine. There, it's CMH. Cysteine, the form is like this: HCOO minus and H3. Yes, science plus VTubing. Let's go. Welcome here, World X. This is Isekai as well, you know? If you just really dig deep, every world we enter is an Isekai. The science is an Isekai, magic is an Isekai, shoujo is Isekai, Isekai is Isekai. Yes. So, cysteine, this is cysteine. NH3 plus and COO minus, and you have CH2 and SH. And this one is methionine. So every amino acid, we know that the top part would be the same. There, that one's the same. The difference is this part at the bottom. So methionine also has sulfur, but it looks like, oh, what's happening? Erase. There you go. Methionine would look like, okay fine, I'll just draw the first part, HCCOO minus, NH3 plus. And then the bottom is like this: CH2, CH2, silver, and CH3. Okay, so that's how it looks like. And then homocysteine kinda looks like cysteine, but at the bottom it's CH2, CH2, SH. So see, if you think about it, cysteine is the top part, normal CH, minus, and NH3, but at the bottom is CH2 and SH. Homocysteine is CH2, CH2, SH, and methionine is CH2, CH2S, and CH3. If you just want to know why sometimes I'm a software engineer student, but I had to learn chemistry as a requirement, and it was very hard for me. I didn't understand most of it, unfortunately. It's okay. We're here, and it slowly but surely will get there. It is so. I just think about it the same as in Pal World, because if you see the other streams as well, like I play Pal World and Pokemon a lot. And then it's like, how can I reach level 60 quickly? Okay, I would need to try and catch a lot of powerful pals, but how can I do that? Then I have to grind and catch like a lot of the less powerful pals. It's just like, okay, I need to level up. And how can I level up in chemistry? It would be grinding the practice problems. That's the life. Organic chemistry really needs you to draw the scribbly, the arrows of where everything goes, and that's how. And sometimes you end up dreaming about all of these too. That's when you know you really are close to Isekai, waking up in the world of chemistry, and that's why you're here. Yeah. But yeah, regarding methionine. Oh wait, regarding the 3 amino acids I mentioned earlier, right? It was cysteine, methionine, and homocysteine. This is essential. Nope, that's non-essential. Non-essential. Methionine is essential. Homocysteine is not incorporated. It's just a metabolic form. And if you want to remember what's the non-essential and essential ones, I think I noted it down here. Oh, thank you. Yeah, it is a lifestyle at this point. Literally, science is a lifestyle. It's lore accurate. Yeah. Regarding, where was it? There's PVT Tim Hall. Well, not pyruvate. If you want to like remember what's the essential and non-essential amino acids, just give me a second. I want to find the page because I also need to like remember which one's which. The last one would be, nah, I can't find it. It's gonna be somewhere there, but just search up this one, like PVT Tim Hall, and you'll see lysine, leucine. It's like that. Those are the essential amino acids. Okay. Two reactions. Yeah, there's also the part regarding the hydrophilic and hydrophobic. So, phospholipids. Okay, what are they? You have a structure of a phospholipid here. It's like this. It's like there's the head and there's 2 tails. I will just draw it in the proper form, just to this one, P-O something, and then O, and then something over here, and then you have the 2 hydrophobic tails. So if the tail looks straight, that is a saturated fatty acid, but if you have a tail that's kind of like this, then that is an unsaturated fatty acid. Because if you think about it, if the fatty acids are like in a layer, then you're like this, but one of them has like a kink, then the next one would be pushed. So that's why you would have these spaces, and that's why it's called unsaturated, because it's literally not saturated. So that's how you think about it. That's like butter, I think. it's like the liquid form of the fatty acids. And yeah, the hydrophilic head is made up of a glycerol backbone and a phosphate group and a polar molecule. And then you have the two hydrophobic fatty acid tails. One is typically saturated or straight, and the other one would be unsaturated or kink, which is also I mentioned, that's the membrane fluidity. Okay, and then you have biosynthesis of it, and the final product would essentially just be the phosphatidylcholine, which integrates into the phospholipid bilayer. Oh wait, I want to change the hat for my mini thing. I think I can change it if I type exclamation point hat. Let me know if the duck hat changed. Yeah, so you have the reaction, like how phosphatidylcholine can release like free choline. It changed. Yes, you can change your hat by typing exclamation point hat. Let's go, because like I have a couple of mini characters there. Next, it didn't for me. Yeah, you have to wait a few seconds, I think. Yeah, there's like a 7 seconds to a few seconds lag time, I feel like. Folic acid, we can talk about the folic acid. The folic acid has, let me erase this first. Here. Oh, your hat changed. I think that's a different hat now. Yeah. Nice. Yes. Yeah, you just have to wait for like 5 to 10 seconds. For the folic acid structure, I won't write the complicated one first. You have the pteridine ring, and the para-amino benzoic acid, and then you have the glutamic acid. I'm not writing the full word because I don't understand what you're talking about, but it's oddly relaxing listening to you. Thank you so much. That means a lot. Like I'm just here to vibe with like science and just like practicing how to yap. Aside from that, I also like do Pal World mostly Saturday evenings, EST. So I think I'm planning on doing that one this Saturday as well. So yeah. Oh, and reminder, folic acid is also, it can be one more. Folic acid undergoes 2 successive reductions, and you use the DHFR, that's the dihydrofolate reductase, and that would use NADPH as a reducing agent. And first, okay, this one I want to write. So here, folic acid, let's start there. Folic acid, and then you have the DHFR, NADPH that's converted into NADP plus, and then you have dihydrofolic acid, another round of the DHFR, and then you have the tetrahydrofolic acid, that's the THF, if you see that a lot of times in the books. And if you notice, the cycle is also linked to folate metabolism and vitamin B12, the one I talked about, because if you want to see, thank you so much, welcome to the stream, Joyce. Yes, science is nice. Okay, next is methyl THF. Here you have, where are the arrows? Okay, here you have S-adenyl methionine, right? And then it gets converted to S-adenosyl homocysteine, and you have the substrate into the methyl substrate over there. But with that gets converted into adenosine and homocysteine, and then you have vitamin B12. That's why people say, always take vitamin B12, don't forget. Yeah, it's important. Vitamins are very important, actually, so don't forget. I always forget. I have to do a better job. But yes. And methyl, you have the different structures and domains of methionine synthase. It's gonna be difficult to draw it out, so if you're interested, you can try and search it up. Wait. Okay, this one is, you have a dual role of folate metabolism and DNA synthesis and DNA methylation. So this is where biochemistry and molecular biology would mix together. Everything is connected. Yes. Okay, everything is connected because in DNA synthesis, folate is reduced to dihydrofolate and then to tetrahydrofolate, right? The folate to DHF, the THF. That's then converted into the 5,10 methylene THF, which will donate to like a methyl to dump DUMP, which would form DTMP, and that's a very critical step for DNA replication. We talked about that before. And DNA methylation, the 5,10-MTHF would be reduced by something which would transfer a methyl group to something, and that's used, and methionine is then converted to like SAM, which is then a methyl donor for epigenetic DNA methylation. So everything is connected. Just don't forget vitamin B12. If you have something to like, if I want you to like take home something from this one, drink your vitamins. B12. Okay. I think we talked about this before. Okay, this one, we have a good map here which would merge everything from here. Okay, so you have methionine is, let's start here. Methionine is activated by methionine transferase to form S-methionine, and that's a universal methyl donor. After it donates it, it becomes S-adenosyl homocysteine, and then you have homocysteine, and then it becomes methionine again, and then you have methyl THF, and then everything happens after as well. So it will just be, again, everything is intercorrelated. Is that the right term for this? All of them just talk to each other. Yeah. Okay. What's the next one? We have a diagram which would outline the biosynthetic pathway from phenylalanine to dopamine, and that is a very, very key neurotransmitter from amino acids. So this is the dopamine that you know, the common dopamine one, and see this where biochemistry and neuro also like collides. It's just one different, like it's just a friend. All of them are interconnected. And yes, phenylalanine, let me just erase this here. Let's see, here you go. So you have phenylalanine, phenylalanine, you know the word, that is converted into tyrosine via phenylalanine hydroxylase, and then you have the tyrosine hydroxylase which would make it into I think this is L-dopa. Yeah, L-dopa. And then you have the dopamine decarboxylase which forms it, which makes it into dopamine. So look at that. See, from phenylalanine you get dopamine. Yes, so it's all talking to each other. And take note, dopamine is again, yes, this is the exact same thing that it sparks up when you watch a lot of YouTube shorts and TikTok, the dopamine rush, and then once it stops, you're like, oh, I need more, and then boom, you just end up watching a lot more YouTube shorts and TikTok. Yeah, and that is the dopamine. It's also using like the same activation pathway, I think, for when you like exercise and stuff, so that's why you feel like happy and refreshed as well if you like work out, or watch YouTube shorts. One of them might be healthier. Yeah. And I'm calling myself out too because I only do the YouTube shorts one. Or YouTube essentially. So yes, that is the same dopamine one. Okay, where is the next one? That's the same. Tyrosine. Oh, okay. Tyrosine, we talked about how tyrosine contributes to the synthesis of tissue proteins. And you also, this one's a good one if we want to clarify this. So you have, you have phenylalanine, right? I can spell this now, alanine. Yes, you have and then it ends up being tyrosine. Okay? So this one is crucial because at some point it becomes tissue protein. And then there's another one. It becomes melanin at some point. And guess what? That's the same thing for when people tell you need it to sleep. Melanin. Oh nope, never mind. That was it. That's a different one. Speaking of shorts, have you considered making science shorts? Maybe that could be interesting. Yes, I am thinking about how to like maybe do it, like maybe bite-sized or just like clip stuff out. But yes, it is in the oven. I'm trying to think about it. And it says what in my head. It's like science, Pal World, anime, Kpop, Kdrama, C-drama, J-pop, and maybe eat. So that's my brain. It's like work, school, sleep, eat, bathe, science. That one. But the shorts, I agree. The science shorts would be a good angle as well. Still thinking about how to implement it. We have tyrosine. Okay, we talked about this. Dopamine, L-dopa decarboxylase, finished. Oh, okay. Now, familiar words might be dopamine, norepinephrine, and epinephrine, okay? So catecholamines are a class of neurotransmitters derived from tyrosine. See? Oh wait, I forgot to write the other three, or that's supposed to be for. Here. You have catecholamines over here. Yes, science media is amazing. It's catecholamines. That one, and you have fumarate and acetoacetate. This one is like it will go into the TCA cycle. So that's a familiar thing, because that is a big cycle. Then after that is ETC, and then boom. So yeah, for catecholamines, you have it is characterized by a catechol group, a benzene ring with 2 hydroxyl groups, and an amine side chain. The major ones, as mentioned, are anime like Dr. Stone and Cells at Work. Yes, Dr. Stone is kind of like science. It's more of like history, I think, and how everything happened. And then Cells at Work has the angle of biology for in an anime form, like biology for like kids. So I like it. It's fun. There, and then we have like K-On! And Sword Art Online. Awesome stuff also. And ReZero. I would like to watch season 3 of ReZero at some point, because right now I'm doing Apothecary Diaries season 2. It's a nice show. Okay, so catecholamines. Does that have an H? Nope. Wrong H. No, erase it. Catecholamine. C. No. C-A. Here. I can spell it. 1. Okay, there. Catecholamines. That is a neurotransmitter class. The common ones are dopamine, the one we talked about, and norepinephrine and epinephrine. Yes, the epinephrine that we know. So dopamine it's in the reward plus motivation and motor control. So if something is wrong there, you get Parkinson's. And then norepinephrine would be for alertness and sympathetic nervous system responses. And then epinephrine is for your flight or fight response. So increased heart rate, blood glucose, and airway dilation. So again, dopamine is the reward system. Norepi would be alertness. Epinephrine is mostly for your fight or flight. So if you feel like running away, think, oh, my epinephrine's going wild. Yeah. And then, deficiencies and dysregulation in the catecholamine pathways are implicated in various conditions such as Parkinson's, ADHD, depression, and pheochromocytoma. That's a new word. Okay. If you want to, the next one would be a good one to draw because it is the catecholamine synthesis pathway. How all of those are made. Hi, thank you for coming. Yes, feel free to just drop by again once you're like free. But yeah, bye Rolex. So for catecholamine metabolism, meta-M, you start with phenylalanine, another phenylalanine, and then tyrosine. So phenylalanine again is, you have the benzene ring and CH2, that is your thing, and then you have CH, NH3, NH2 plus, and then COO minus. Okay, but yeah, you have tyrosine, not this one there. After tyrosine, you have dopa. So phenylalanine, tyrosine, L-dopa. Dopamine, okay? After that, you have norepi, and then epinephrine. So it's just in order. Again, phenylalanine, tyrosine, dopa, dopamine, norepi, epinephrine. Yay! And 4 different cofactors or how they happen. Vitamin C, because from dopamine into norepi, you need vitamin C here. And oxygen. So that's why people say like eat oranges and also breathe air, healthy air, smell the trees, touch grass, because you need those to feel alert, because norepi is for alertness. So far, vitamin C and B12, crucial things. Okay, we talked about that one. Tryptophan. Oh, there is also another thing. It can be converted into tyrosine as well. I mean serotonin. So that's another one. You have tyrosine synthesis, catecholamine synthesis, and serotonin synthesis. All of those important stuff. And then you have the neuron, glutamatergic neuron. It would recycle the glutamate through glutamine. So after it gets released, I think this one we have to write. Like it, because it involves the synaptic cleft, which might be more familiar in picture form. So if this is the astrocyte, and you have the presynaptic terminal, and then you have the postsynaptic spine. So you have glutamate, and then glutamine, GLN, goes through here. You have GLN to glutamate, and then boom, it travels there. So that's how they all talk to each other. So again, after the glutamate is released into the synaptic cleft, excess glutamate is taken up by astrocytes via the glutamate transporters, and within it, glutamate is converted to glutamine. And then in the neuron, glutamine is converted back to glutamate. Well, voila. So another talk about glutamine catecholamine biosynthesis, but we talked about that. You have the different terminals, right? The dopaminergic terminals, noradrenergic terminals, and adrenergic terminals, because that's adrenaline is kind of the same thing as like epinephrine, so that's why it's also called noradrenaline and adrenaline, I think. Yeah, correct me if I'm wrong, but I feel like that it is that one. Oh, and then going back into the liver and muscle, here, liver and muscle over here. Okay, so in the tissues, we know that it becomes from pyruvate into acetyl-CoA, right? That's a familiar thing. Pyruvate to acetyl-CoA, pyruvate to acetyl-CoA. And then the acetyl-CoA could go into the TCA cycle. And remember, from you also have acetoacetate becoming the acetyl-CoA. So yes, the ketone bodies may block the entrance of fuel into the TCA cycle, because if you have a lot of ketone bodies such as acetoacetate, this one would lead to increasing the amount of acetyl-CoA, which would then inhibit pyruvate becoming the acetyl-CoA, because the body then thinks that you have a lot, and this one will be like inhibited too, I think, because it'll be like, I don't need more energy, so that's why. And to adapt, cells increase glycolysis to generate ATP from glucose directly. Yes. And now to just have a mini review of amino acids. So can you describe the major metabolic fates of amino acids and their significance beyond protein synthesis? Okay, so amino acids, although wait, let's erase it here, just to write the structure of it. So again, amino, not red, let's make blue. Amino acid like this, COO, and then you have NH something, was it like 2? And then you have like H and then R. Let's just double check, because it depends on the pH. Here. Oh, I found the PVT Tim Hall page. It is phenylalanine, valine, threonine, and all of that. Yeah. If I'm using this pH, then it is H3 and plus. Nice. Okay, so yeah, amino acids, although commonly known as the building blocks of proteins, they play a far more dynamic roles in metabolism, and they could serve as different precursors. So again, if we want to describe the different fates of amino acids, they serve as precursors for like neurotransmitters, hormones, and purines and heme groups. And during fasting or stress, the carbon skeletons can be oxidized for energy, which could possibly like enter the central pathways such as glycolysis and the TCA. So again, what are amino acids for? They're known as the building blocks of proteins and could also serve as the building blocks for hormones, neurotransmitters, proteins, purines, and heme groups. So why is glutamate considered the central hub in nitrogen metabolism? And how does it facilitate nitrogen disposal? So glutamate is a collector and distributor of nitrogen in amino acid metabolism, because usually if you have transaminases, amino groups from various amino acids can be transferred to alpha ketoglutarate, which would form glutamate. Okay, and then that centralization allows nitrogen from multiple sources to be funneled into just one thing, a single metabolite. So that's why. So again, you have a couple of amino acids into alpha-ketoglutarate, into glutamate, and then if you have glutamate, it would undergo, yes, dab. Welcome back, George Rojas. Thank you for using the dab one. That's awesome each time. Yes. And then, after you have glutamate, it will undergo oxidative deamination via glutamate dehydrogenase. And then you can form the free ammonium and regenerating the alpha-ketoglutarate. So just going back cycle so that you need them to just keep on going. And what's the form of PLP? The pyridoxal phosphate, and how does it enable transamination reactions? Okay, how? PLP is an active form of B6. It also serves as a coenzyme in amino acid metabolism, especially in the transamination decarboxylation and elimination reactions. So again, what are the different major metabolic fates of amino acids? They mostly known as building blocks of proteins, but then they are also building blocks for neurotransmitters, hormones, proteins, and heme groups. And their carbon skeletons can be oxidized for energy, which would enter central metabolic pathways. Why is glutamate considered the central hub? It's because from glutamate, from different amino acids can be transferred to alpha-glutarate, and then alpha-glutarate would form glutamate. And this centralization allows nitrogen from multiple sources to be funneled into a single metabolite. Ooh, I'm gonna try and change my hat. If I'll type exclamation hat, hopefully it will change. I think mine is the wave one right now. Will it change? How many seconds? Nice! It's now the sparkly one. Awesome. Okay, so the ammonium finished. We finished that one. And then how does the glucose alanine cycle coordinate nitrogen disposal and energy metabolism between muscle and liver? Okay, so we talked about this before. In the skeletal muscle, especially under aerobic conditions, pyruvate accumulates from glycolysis, right? And then, to manage the nitrogen waste, glutamate donates its amino group to pyruvate via ALT, and then that would form alanine. Yes. Oh yeah, if you do the exclamation point hat, I think after it changes the hat, you need to wait for a few seconds before it changes. Oh, I think yours changed into the chef. Yeah. So again, under anaerobic conditions, pyruvate would accumulate from glycolysis. To manage nitrogen waste, glutamate donates the amino group to pyruvate by ALT, and that would form alanine. Why is ammonia toxic? Ooh, it's like Rosé's song, it's the toxic till the end. And ammonia is we know that ammonia is a small uncharged molecule that readily crosses membranes, and it can disrupt proton gradient in the mitochondria. And in the brain, ammonia leads to astrocyte swelling. It would deplete your alpha-ketoglutarate, and it interferes with glutamate and GABA signaling. Everything awful. So to avoid toxicity, the liver would rapidly convert ammonia to urea via the urea cycle. And that's when you have the carbonyl phosphate that we talked about earlier. So just remember, ammonia bad. Ammonia bad. It's bad. So if you are in the lab, try to not smell it if you're working on it. Okay. That's why I wear a mask if you need to work with it. Okay. This one's an experimental question. How would you experimentally determine if a transamination reaction has taken place in vitro? So one approach would be to use a radio-labeled amino acid and incubate them with alpha-ketoglutarate, and also a specific and a specific aminotransferase. So, if a transamination occurred and we put in a radio-labeled amino acid, a radio-labeled glutamate will be produced. So that's when you know it happened. But how can you see? It will be detected by a TLC, a thin layer chromatography, mass spec, or HPLC. So just start with radio labeling an amino acid, throw in alpha-ketoglutarate. How can we demonstrate that PLP is essential for a particular transaminase reaction? You could perform parallel in vitro reactions, one with aminotransferase and PLP and the other one without PLP, and of course you'll see a difference. Add glutamate and pyruvate to both, and then after that just monitor the production of alanine and alpha-ketoglutarate. So yes, that's how you do it. And what are amino acid oxidases and how do they differ from transaminases in amino acid catabolism? Well, amino acid oxidases will catalyze the oxidative deamination of amino acids with using two things that we know, FAD or FMN. So you'll always see those. What makes the hydrophobic effect thermodynamically favorable in aqueous environments, and how does it influence protein structure? So the hydrophobic effect, we talked about that. It's driven by the entropic cost of organizing water molecules around non-polar substances. So I will just draw it again. Where is this one? It's here. So if we draw it out, the best way would be here. Where is it? My marker. Found it. So if you have the cell membrane here, and all of the hydrophobic, I flipped that. All of the hydrophobic tails would be together, and then the hydrophilic heads would just be facing the water. So that's how they keep it organized. Water is around it. Okay, we have the shift? Okay, what determines whether an amino acid is glucinic, ketogenic, or both, and how would you determine it? So it is, if you can check depending on the fate of its carbon skeleton after deamination. Okay, so glucogenic amino acids would generate pyruvate or TCA, while ketogenics would generate acetyl-CoA or acetoacetate. So again, glucogenic would go into the route of pyruvate or TCA, meanwhile ketogenic would go into the route of acetyl-CoA or acetoacetate. And then, again, if you want to experimentally test that, radio-label the alpha carbon of an amino acid and just trace how it is incorporated into the glucose or ketone bodies. And yeah, radio-labeled CO2 production could also indicate TCA cycle oxidation. And how does electronegativity influence the behavior of functional groups in amino acid side chains? Electronegativity would affect electron distribution within chemical bonds. That would affect its polarity and reactivity. So again, electronegativity affects the electron distribution within chemical bonds, and then if you affect the chemical bonds, then the polarity and reactivity would be affected. So, for example, the hydroxyl group in serine is polar due to the electronegativity of oxygen, and that will allow it to participate in hydrogen bonding and catalysis. And in contrast, nonpolar side chains, like those in leucine or valine, have minimal charge separation. So that's how the different protein folds would fold and interact. It will also explain, electronegativity also explains how the residues are stabilized. Can you explain the nucleophilic addition mechanism of nitrogen-containing nucleophiles to carbonyl groups, and how does this relate to transamination? So, nitrogen nucleophiles like amines attack electrophilic carbonyl carbons, such as like in aldehydes or ketones, and they would form a tetrahedral intermediate. Okay? In acidic conditions, this intermediate can then lose water, so you'll form the Schiff base. And then I think the next part is like a couple of questions about the urea cycle. Just bear with me here. It's just like about a few questions. Now, what is the overall purpose of the urea cycle and where does it occur? Urea cycle detoxifies ammonia because ammonia bad. It will convert it to ammonia. It is a less toxic molecule excreted in the urine, and this cycle occurs in the liver. So again, urea cycle would detoxify ammonia, converting it to urea, and then you get rid of it in the urine. It happens in the hepatocytes and the liver, both in the mitochondrial matrix and the cytosol. It's the primary mechanism of what? For nitrogen waste disposal. What is the rate limiting step in urea? And what activates it? Rate limiting step is CPS1, the carbamoyl phosphate synthetase. And then it is activated by NAG, the N-acetyl glutamate. How does ammonia from the tissues reach the liver? Ammonia is transported as glutamine and alanine. Glutamine synthetase in the tissues incorporates ammonia into glutamate to form glutamine. So again, you have ammonia into glutamate into glutamine. Meanwhile, in the muscles, pyruvate accepts the amino group from glutamate to form alanine. So that's why alanine and glutamine is the right answer. Those both would then travel to the liver, where glutaminase and alanine aminotransferase would be. Okay? Then describe the reactions. That's just the CPS1 catalyzing the formation of carbamoyl phosphate from NH4 and CO2. So mitochondrial reactions, you have the CPS1 catalyzing the formation of So okay, let's start. NH4 and CO2, ammonia and CO2. Write an arrow. I can write an arrow. Actually, why am I just talking? I can actually write it out. Right here. Erase. There. And I will use the black ink. NH4 plus CO2. Carbamoyl phosphate, and then this is CPS1. Nice, huh? And then after that, it reacts with ornithine, and OTC would help that out. Okay, cytosolic steps of the urea cycle, because we mentioned it's like the mitochondria and it happens in the mitochondria and cytosol, right? Cytosolic steps would be, in the cytosol, citrulline would combine with the aspartate, and that is catalyzed by arginosuccinate synthetase to form arginosuccinate. It is then cleaved by arginosuccinate into fumarate and arginine, and arginase, then blah blah blah. So yeah, that is the flow. What is the source of 2 nitrogen atoms in urea? 1 nitrogen is from ammonia, other is from aspartate. So again, you have ammonia N, aspartate N equals overall urea. How is it connected to the TCA cycle? You have shared intermediates such as fumarate generated in the urea cycle, and aspartate is derived from oxaloacetate. What happens to the carbon skeletons of amino acids after the nitrogen is removed? After deamination, the carbon skeletons would then become alpha keto acids, which would again enter the TCA cycle. How can we confirm if CPS1 is the rate limiting step? Well, just prepare liver mitochondria, measure CPS phosphate reaction in the presence or absence of NAG. We mentioned NAG is the activating form. And But if it doesn't happen, if you don't have NAG, then you know that it needs NAG. How does glutamate act as a safe ammonia transporter? In the peripheral tissues, the glutamine synthetase would catalyze the ATP-dependent incorporation of ammonia. We talked about this earlier. Describe the glucose-alanine cycle. In the muscle, pyruvate from glycolysis is transaminated to alanine. It would enter the bloodstream. It's taken up by the liver. It would donate amino group for urea synthesis. An amino group. What role does glutamate play in nitrogen metabolism? It's a carrier and a donor. What happens if you don't have OTC, or the ornithine transcarbamylase? Okay, so this one, it disrupts the second step of the urea cycle, but it would prevent the formation of citrulline. And then you have hyperammonemia, because the ammonia cannot be efficiently incorporated into the cycle, so you just have a lot of ammonia in your body, and that would present as lethargy, vomiting, cerebral edema, a coma, and especially during stress or high protein intake. How is methionine linked to methylation reactions? It's converted into SAM, or S-adenosyl methionine, another universal methyl donor. How is methionine regenerated from homocysteine? Oh, this one's good. We mentioned that there's a reaction that requires vitamin B12, and that would ensure the continuation of the methylation cycle. What is the methyl trap hypothesis? Well, it's just when you don't have enough vitamin B12, the methionine synthase activity is blocked. Then you have a lot of it cannot be recycled back into THF. You have folate deficiency. What enzyme converts serine to methylene THF? That is the serine hydroxymethyltransferase. That's a handful. That word is long. Okay, I think. Did we talk about all of them? Yes. Yes, we did. Thank you for watching. I think that's all for today about the urea and its friends. But yes, that is all for now. Thank you for watching. Bye bye.